Good morning, good afternoon, everybody. We are very pleased and welcome Professor Günther Zeck. Professor Zeck is a professor of biomedical electronics and systems at the Technical University of Vienna. He completed his PhD in physics in the Technical University of München. He conducted research on neuroelectronic interfacing in the Max Planck Institute for Biochemistry under the supervision of Professor Fromherz. He then joined the Massachusetts General Hospital in Boston as a postdoc in the Department of Neurosurgery, and he studied interfaces with healthy and blind retina with microelectrodes array. Between the years 2010 and 20, he directed the research group in neurophysics at the Natural and Medical Sciences Institute at the University of Tübingen. His group, in collaboration with some other partners in the University of Tübingen and Dresden, developed new strategies for artificial vision restoration, ranging from novel electrical stimulation protocols to optogenetics, cell transplantation, and pharmacological intervention and protection. In 2020, he appointed as a professor of biomedical electronics at the Technical University of Vienna. His main research topics includes microelectrodes array in life science, development of stimulation strategies for artificial vision and restoration, and large-scale extracellular and electrophysiological recording and analysis. And we are very excited to have you with us here in this vision seminar of Bar-Ilan University. So please, Günther, thank you.
Thank you very much for this very nice introduction. It's a great pleasure to be here and to have the opportunity to share with you results that myself and my team obtained in the last years in assessing vision restoration using ex vivo retina, and the goal is, of course, to improve vision restoration. Here is a brief outline of my talk. I will motivate first why we do the research and also share with you some thoughts and some results on our experimental setup. And I decided to talk today only about electrical stimulation because that is something where I work the most time and which I personally find very interesting. But of course, I and others have to admit there are other stimulation strategies like optogenetic stimulation, which might eventually be competitive and potentially even better. The motivation is pretty simple, but worth to be spelled out clearly. So retinal implants cannot be considered a success story yet, I would say this, even though myself, together with probably many here in the audience, strongly believe in the technology. So the reason why I'm saying this is that the retinal implants still show a mixed performance as reported by patients. So most patients report a resolution which is below legal blindness or marginally better, considering new implant results from France. Furthermore, several patients also in the past reported fading of their visual percept. So even though they had a visual percept evoked by an artificial implant or some artificial stimulation, this percept was not constant, but it fades. So these are more or less the two major motivations why we started research, or why we performed the research I will talk today about. Here are some figures and photographs showing the experimental setup we use in the lab. On the left, you should see the pointer. On the left, there is a schematic retina with the ganglion cells. Some of the ganglion cells are shown here in green, These cells express an optogenetic actuator, which will be important in the second part of the talk. The activity of the ganglion cells in the ex vivo retina is very well recorded with microelectrode arrays. This is a very common technique in many labs worldwide. I will show you some examples which I find very interesting. If one goes to a very high density of the electrode arrays, one is able to obtain electrical imaging, not only of the activity in the ganglion cells, but also the axons, which is an important aspect for stimulation. Then you probably all know that in addition to the ganglion cells in the retina, there are bipolar cells or layer of bipolar cells. There are interneurons, amacrine cells, which are just schematically shown here small. And there might be some remaining photoreceptors depending on the age of the degenerated retina one is investigating. So the tools I'm using, or we are using, are mostly CMOS-based microelectrode arrays. We recorded mostly from the ex vivo mouse retina. We had a chance several years back in Tübingen to also record a primate retina. This chance is over at the moment. We are able to apply electrical stimuli, arbitrary electrical stimuli, and simultaneously record the activity. And we are also able to optogenetically stimulate, so to apply photostimuli and simultaneously record. Almost all the results I will show you today have been published very recently by Andrea Corna on electrical stimulation and by Miriam Ray, PhD student in my lab, on the optogenetic stimulation. Before coming to the results, I want to share with you one more slide showing this identification of retinal ganglion cells and axons on a high-density CMOS array. What you do see are two almost identical figures, color-coded, are the positions of the ganglion cells. In this panel, they are in white. In the other panel, they are in black. These positions of the ganglion cell somata are obtained after a procedure called spike sorting, which I will not talk today about, but just for you to know that there is a mathematical analysis from the recording to this drawing here to this figure. In addition to the position of the cell somata, we are also able to identify the axon paths. And you probably all know that in the retina, the axons of the ganglion cells project towards the optic nerve head. These images are obtained just from electrophysiological recording, so there is no fluorescent imaging or no histology going on here. You will see in a movie in a few seconds how this axonal action potential propagates, and will hopefully understand then better how these images are generated. Now let's come to the first question, which is a rather simple one regarding the temporal resolution or the fading. We started with pulses of 1 millisecond duration, biphasic pulses, and applied them at the frequency of 25 Hertz to the AD10 retina and recorded simultaneously ganglion cell activity. You do see here the artifacts, and however, you also do see spiking. So I selected one trace which was not artifact cleaned, and what is hopefully very obvious that after 5 to 6 stimuli, the response of the ganglion cell disappears. We repeated this stimulation with many other cells, and on average with 25 Hertz, with 40 Hertz. So the blue symbols are for 25 Hertz, red symbols are 40 Hertz. And we always obtained more or less the same result, that after 6 to 10 stimuli, the activity of these retinal ganglion cells cannot be modulated further. This fading has been reported previously for other ex vivo retina preparations. It has also been reported for patients wearing retinal implants, to my knowledge, for a subretinal implant by Katharina Stingel's group in Tübingen and Ibrahim Srener's group. And for these pulse durations of 1 millisecond, I have to mention that we did not test all the different pulse durations that one could imagine. So, and I'm aware that like the group of E.J. Cicilniski in Stanford and the group from Andreas Hirlemann in ETH Zurich, they use much shorter pulses, which apparently seem to avoid this fading issue. However, we decided when we started the project to use smooth waves or sinusoidal stimuli, which are known to activate retinal ganglion cells. So that's the shortcut RGC, retinal ganglion cells in equiretinal configuration. And one of the first studies, to my knowledge, comes from Shelley Freed's lab from Harvard University. He published in 2010 a paper where his group stimulated retinal ganglion cells, and they recorded in the rabbit retina with patch clamp electrodes, and they identified spiking in every phase here of this 5 or so repetitions of the sinus. So this was for us a starting point, and we decided to repeat this or to repeat this stimulus pulse, however, to investigate it in depth and to also address specific question. Before going to the question, I would like to share with you a short movie. What you do see here is in gray the entire CMOS-based microelectrode array, and every pixel represents the voltage on one sensor. And if I start the movie, you do see 4 black squares flashing up and down. This is the stimulus, which was not completely removed here on purpose, so that you do see it. And what you should also see is an action potential generated by a cell, and here is a cell position, and the axon path is also visible in this movie. The movie is generated by averaging multiple spikes, but because the action potentials are very reliable, we can identify the axonal spike as well. I would be happy to talk about this later. So let's go to the results from the RD10 retina, from the continuous stimulation, the continuous application of sinusoidal stimuli. And on the upper graph, you do see on the upper graph the sinusoidal starts. That is also recorded. And after appropriate high-pass filtering mainly, we identify the single spikes which are random before the stimulus starts and very reliable in phase with the sinus after the stimulus starts. This was done for one certain electrode configuration. We investigated different electrode sizes. On the y-axis, here the lowest is the smallest electrode, which increases with as the y-axis goes up. And we quantified how well the cells can be activated using a so-called reliability index, which measures in how many repetitions out of all the repetitions there is at least one spike. You do see here for several examples that we have a very high reliability even for this retinal ganglion cells in the RD10 retina. There are some cells which show a low reliability for small electrodes, and this occurs because our electrodes are at a certain distance to the cell. We repeated these experiments with different preparations in wild-type retina, in primate retina, in RD10 retina, and the average results are a very high reliability. And given that we are aiming for an as high frequency as possible, we decided to perform all the following experiments only with 40 Hertz, because we are going to investigate now several stimulation experiments. So the first one is how well can we discriminate two stimuli? And the stimuli are defined as following. So we have stimulus 1, which is defined by a square of about 120 by 120 micron. Then we shift, so this is an electrical stimulus. Then we shift the stimulus to position 2, and so the shift is 32 micrometer. So stimulus 1, that is a spatial scale, and the temporal scale are 4 sinusoidal stimuli. And for stimulus 2, again, 4 sinusoidal stimuli, but shifted in time. And on the right, you see the results we obtained. So every circle represents 1 retinal ganglion cell, and the color code represents the change in activity. So cell response is not the absolute firing rate, but the change in activity compared to the condition where there is no stimulus. All these details are described in the paper by Andrea Corna, which I didn't put on every slide. And the question is, can we separate based on the activity of these 2 stimuli? Can we separate or tell apart, discriminate the 2 stimuli? Before telling you the result, I would show some intermediate results. So first of all, I want to emphasize a little bit more the spatial selectivity of sinusoidal stimulation. Here we selected, or I selected 2 cells marked by 1 and 2, and the cells were stimulated with electrodes of different size. The smallest one being this black one, then we have a blue, a purple one, and even a large field. Depending on the stimulus intensity, indeed, as mentioned before, we can evoke reliably activity in the retinal ganglion cells by sinusoidal stimulation with the blue square shown here or with the purple one in cell number 1. And if we change the stimulus amplitude, we obtain conventional or stimulus strength relationship, stimulus response relation. What I find interesting is that for cell number 2, which is farther away and which has an axon crossing over the stimulating electrodes, there is no activation at all. So the cell maintains its spontaneous activity. However, the cell can be stimulated, as shown here with this dashed line, or symbols connected by the dashed line, if we apply a pretty large, or if we stimulate with a pretty large electrode of 1 square millimeter in this case. So this is a first indication. However, we then went further and investigated more than 1,000 retinal ganglion cells stimulated here by 1 very well-defined electrode, which is shown by this white dashed line. And again, the color code represents how the cell responds, so how much the cell is activated, increases its activity compared to spontaneous activity. And we evaluated this for over the distance from the electrode center, and the data can be very well fitted by a Gaussian. So only, and the sigma of the Gaussian is 57 micrometer. So only within a very narrow range from the electrode center, we activate retinal ganglion cells in RD10 retina. Even though there are probably several ganglion cells here with axons crossing the electrode. And we looked, or Andrea looked for now specifically for those cells which indeed had their axons crossing across the stimulation electrodes. Stimulation electrodes are shown just for better visibility here in yellow. And for none of these cells under our experimental conditions did we activate some action potentials in the axons which would be visible in the cell soma. This was true for 2 different experimental conditions that we investigated. So,
Günther, can I ask something?
Sure, yes.
So you have this electrical stimulation, and I guess that you have an artifact, and you probably remove it, So you can only see more later delayed action potential. So what is the window that you remove?
Yeah, that's thank you for the question. I forgot to mention, we did not remove any window. I had this, if I go back, yeah, here you would see there is a very small artifact here at the beginning, but that doesn't need to be removed, and there is really spiking going on from the very first sinus. And the reason is because we use smooth waves. So the smooth waves create an artifact in the electrodes, but using high-pass filters, we can remove it completely without having any window. Yeah, that's a very good point. I forgot to mention this because that is important. Also, this direct activation, the spikes in the retinal ganglion cells occur usually within 1 millisecond after the stimulus goes on. It doesn't seem to be the case for the sinusoidal. We don't know exactly at the moment why. So it's not when we start the sinusoidal, of course, there's a very smooth waveform, and the first spike is not within the first 1 millisecond. I will come to that later. Yeah, thank you. So let's see the result. Coming back to the discrimination of the two stimuli. Here is, you have already seen this slide. So the question now is, if we have Gaussian or distributions of selectivity with a Gaussian of 57 micrometer, and we separate the two only by 32, will we be able to discriminate or not? And for this task, we collaborated with the group of Jakob Macke, who is a theoretician in Tübingen, and they used logistic regression, so a well-known model where they trained a model using a certain number of stimulus repetitions, and they then predicted the stimulus from novel stimuli. So maybe I should rephrase this. So we repeated stimulus 1 and stimulus 2 50 times randomly, and based on known presentations of about 70 or 80% of the stimulus repetitions, they trained their model, and they predicted the remaining 20 or 30% of the stimuli using the model. And depending on the retina, this is also a very interesting result, depending on the retina, the prediction accuracy varied. So here there are displacements shown for 32 micrometer, and every gray bar represents one single retina preparation, and every colored bar represents the merged retina. We called it the merged retina because we were able afterwards, after the recording, to pool all the ganglion cells and to create so-called artificial or merged retina. And the goal was to see if we can increase accuracy, which was for some of the stimuli indeed the case. The second motivation is, of course, that in some of the preparations, the number of cells that are recorded and then also activated is smaller than in others. So on average, for rather large stimuli, even with a small displacement, the stimuli could be discriminated with a high accuracy of up to 90%. If the stimulus itself is small and the displacement is small, the accuracy drops. We were interested also to learn if all the cells are required for the model. So if all the spiking of the retinal ganglion cells is important, and it turns out not. And this is, of course, also somewhat associated with the model itself. So it appears that only 10 to 20% of the population of every retina is important. And not surprising, it is that population that is also activated. So if we feed into the model 100 cells, but out of these 100 cells, only 10 cells are really strongly activated. These 10 cells are important for the model to help discrimination. This is not so surprising, but it's definitely good to see that the model behaves as predicted, that after 10 to 20% for every retina, the upper accuracy is achieved. We started also another type of experiments, which is definitely not complete yet, but we were interested to learn if also contrast can be discriminated. So we applied the following stimulus sequence. Stimulus 1 was also kept at a constant stimulus amplitude, and for stimulus 2, we decreased stimulus amplitude, so to artificially create a contrast. And depending on the electrode size or the object size, we could achieve higher or a pretty high accuracy of 80%, and for 100% contrast. So when the stimulus obviously disappears, the accuracy goes to 1. I have to say this is, of course, a somewhat mixed experiment, and we need to continue on this to come to a real solid conclusion, because also the stimulus has a certain size, and the results, of course, depend on both on contrast and stimulus size. So it is not a clear-cut result here only for contrast. But it is encouraging, though, that there is, even with low contrast levels, can be discriminated. Now, I would like to come a little bit more to the question, why is it possible at all to discriminate with epiretinal stimulation these two nearby stimuli? Or why do we believe that we activate ganglion cells? When I started the project, I thought that we will activate the network with sinusoidal stimuli, and I think the majority in the audience would say that low frequency stimuli mostly activates the network. We were surprised when we applied, so this is when we applied pharmacological blockers in RD10, we didn't see much of a difference. And this is one result supporting this statement. Again, on the left in RD10, there are the retinal ganglion cells stimulated, and above the electrodes, there is a high cell response. Outside, there is a low cell response. And again, we can approximate this decay very nicely with the Gaussian fit. We applied to these retina glutamate receptor blockers, which blocks the excitatory input to the ganglion cells, but could not see any significant difference in these retina for these ganglion cells. So we have the same decay as before. The picture or the result looks different in wild-type retina. So in wild-type retina, initially, with the very same setup, the same stimulation protocols, the decay was not as steep as in the RD10 retina. We even didn't fit the Gaussian here because the fit quality was bad. And you see on the x-axis, the decay goes from 1 to, I would say, to 500 micrometers. So it's not as spatially selective as in RD10. When we applied DNQX and AP5, the situation in the wild type retina changed, and indeed there are some cells which still can be activated by this stimulus, but most of them not. So we have restriction, or again, a spatial selectivity in wild type only after application of the pharmacological blockers. And the third, a second support that the synaptic blockers do not change the activity in RD10 is shown here. So here are again two stimuli, sinusoidal stimuli for phases. You do see in every phase activity in this AD10 cell. When we apply the synaptic blockers, the activity remains and it is still there after the rewash. In spontaneous activity, this is just a control that indeed the blockers work. So when we have spontaneous activity in AD10, there is this well-known bursting activity, which occurs at different frequencies depending on the age and so forth. And after synaptic blockers are applied, the oscillating activity disappears. So the synaptic blockers indeed worked. That was also a certain concern. And when we rewashed the retinal sample, this bursting activity returned. So we are confident that in the experiments that we conducted on the adult AD10 retina, so the age is reported in the paper, I think it's about 150 days, we do activate directly the cells with sinusoidal stimuli. I would like to share one, so this is, I would say, a very interesting result, to a certain degree also unexpected for us. But the question is, can this be implemented at all in a retina implant? Because to my knowledge, most retina implants work with pulses because pulses are easily to be implemented in hardware. So we collaborated here with Professor Rothermer from the University of Ulm. We published a first conference proceeding, and they were able to build electronic circuit which is can be used as an efficient electrode driver for sinusoidal stimulation of retinas. So I would like to come to an interim summary. What are benefits of sinusoidal electrical stimulation? That they reliably activate retinal ganglion cells in AD10 without axonal activation. The discrimination of two objects by 32 micrometer can be achieved with high accuracy. There is, however, a large variability in the prediction accuracy between the retinal samples. We did not test if that, what are the reasons here? There are multiple possible reasons. I did not talk today about stimulation thresholds, so how much we would need to apply, how much charge. We think that the charge threshold density is much lower. We have all the numbers published. However, one major drawback, of course, is the stimulation is very cell type unspecific. So it's well known that there are in the retina at least these two major channels of on and off retinal ganglion cells which are not activated. Therefore, we collaborated with a group of Philipp Behrens, again from Tübingen, and asked them if they could modify the sinus such a way that it activates mostly off and not on cells. And because they had a very good model of an off bipolar cell, they came up with this stimulus waveform, which was published last year. And they suggested that with this stimulus waveform, indeed, mostly off bipolar cells are activated. And here is the cumulative release, so the vesicle release from bipolar cells, not the spiking activity from ganglion cells. We don't have results yet on the spiking activity from ganglion cells. Another search for alternatives to sinusoidal stimulation we also published last year was to investigate white noise stimuli and to look, maybe we find different filters, temporal filters from white noise stimuli. However, in the AD10 retina, all the filters were very, very similar. So we could not distinguish between the different cells. So in the last part of the talk, I would like to show some recent results on optogenetics as an alternative. I forgot to ask how long the talk should be. Is it 45 minutes or am I already over the?
No, it's okay. You have 50 minutes for the talk and then questions.
Okay, I'm fine. Thank you. Thank you. So I would then continue with the talk. Of course, electrical stimulation is very attractive. It has a long tradition, I would say. There is already a huge knowledge base. There are also implants which have been commercialized. Others are in progress of being commercialized or in clinical trials. However, there is at least a second stimulation strategy which needs to be considered, and there were already talks in this seminar series by Botan-Roska, Serge Picot, who investigate optogenetics. And we asked the question, does photostimulation or optogenetic stimulation offer or higher temporal and higher spatial precision? And of course, is it possible to achieve cell type specific activation? So this was a PhD project of Miriam Reh, and she used the same CMOS-based microelectrode arrays, and she used mouse retina expressing channel rhodopsin in parvalbumin positive retinal ganglion cells. And that mouse retina was again crossed with an RD10, so the photoreceptors degenerated. Here, and she applied patterned optical stimuli from DMD, so digital mirror display. In panel B and C, excuse me, there are some micrographs showing this green fluorescent ganglion cells on the microelectrode array. So the pictures are not beautiful because we performed these images after the recording. If one stain and not on fixed tissues, so if one stains the retina, these are usually the images one obtains. So what is the first challenge when using or combining optogenetic photostimulation with recording, with microelectrode recording? The photostimuli are usually of high intensity and they may create artifacts. So they might damage the chip, or they might create a strong light artifact. And this is indeed the case for some of the coatings of the chip. Other coatings show a much less light sensitivity. And second is indeed that when the optogenetic stimulus starts, there is an artifact and which might bring the whole chip out of working range. And we circumvented this by adding a repetitive sensor reset. So I don't know if this is true here, the number, but somehow every 100 milliseconds, or that it's adjustable from the chip, one can basically blank for 200 microseconds the recording. All the numbers are reported in the paper, so I might miss here some numbers, but so for a very short time, one blanks the recording, one brings the sensor back to the working point, and then recording can continue. And this is how the traces look like. So we miss some 100, 200 microseconds of the recording, potentially also some spikes. So we asked again the question on spatial localization. Miriam decided to use slightly different stimuli, so therefore the two discrimination tasks don't match perfectly. So she decided to use bar-like stimuli first, and also to map the receptive field of these photosensitive ganglion cells. And here are two cells shown with kind of expected receptive field maps. And the maps also depend on the stimulus intensity. So this was not unexpected. We then also asked, do we activate axonal spikes? And this is shown for cell number 2. So cell number 2 is shown in the background, this dark gray let's call it footprint of the cell on the electrode array. So these are the sensors which record activity from the cell. And then Miriam presented bars perpendicular to the axon, and she recorded at the soma position. And indeed, it seems that even bars which are farther away, so where the axon is, evoke some activity in the ganglion cell. So the question is, does this hurt the spatial resolution? So again, using logistic regression, she discriminated stimuli. In this case, I think it's again the same value of about 30 micrometer. She presented individual stimuli, very narrow stimuli, and asked how well she can discriminate. And she was able, with this logistic regression approach, to discriminate even, I think this is 30 micron. Apparently, I lost here the label. Yeah. But we were surprised that optogenetic stimulation seems to be as good, even though we activate axons. So then we went to, or Miriam went to investigate a second stimulus, which consists of alternating grating. So she applied these stimuli A and B, where the positions of the bar are alternating in phase. And in the upper graph, that is kind of the control. She didn't apply any spatially structured stimulus, and indeed there was no significant change here. And of course, she trained the model, but the model accuracy was 50% or over. So we have a certain confidence that our model works well. It doesn't predict anything wrong. And then she went on with 20 micron and increased here. I will show in a second the summary slide. So she changed the grating width, and with very few neurons, she was able, using this logistic regression approach, to discriminate gratings. This is a final result. So we had here is a grating width from 0, that is baseline 50%, and already at 10%, 10 micrometers, excuse me, we have a pretty high prediction accuracy. I have to say this prediction accuracy always has to be understood that we train the model with 80% or 70% of the repetitions, and then we just predict the remaining test samples. So this has to be kept in mind. Here is just a summary comparing our results to previous work at the moment when we submitted the paper, so there might be even more in the meantime. So there has been reported a pretty high spatial resolution given in cycles per degree for non-human primate retina. I cut here for visualization purposes the references, but they can be found. And for mouse retina, it slightly decreases. And for our results, we obtained with logistic regression resolution of 1.7 cycles per degree, which is even higher than one would say normal vision in healthy mouse retina, which is to a certain degree surprising. So therefore, we went.
Can I ask something?
Sorry?
Can I ask something regarding this?
Please, yes.
Yeah, so again, so if you think that the model can discriminate 10 micrometer and well, the resolution, as you were pointing in the red, the receptive field is about, in the mouse is about 200.
That's correct.
Yeah. So this is like a sub field resolution. What does it mean that the model can predict it? What is the physiological relevance of this? And maybe, you know, some cells are firing, some cells are not, but does this have any physiological relevance?
Yeah, that's a very good point. And so when we go back, that is indeed, so the model needs the input for those cells which change activity. That means the brain would need to learn to listen to those cells which change the activity. If that is not, I mean, with in vitro experiments, we cannot answer this question. But if the brain would not be able to learn from a restricted, it doesn't need to listen to all the cells, but it needs to listen to 5 to 10 cells about the change in their spiking activity. That is what would be the physiological translation of this statement. Therefore, I mean, using the model, we obtain a pretty high value, but it is indeed a very open question if that is relevant for the translation in vivo. However, and I would like to show you one last slide, using another approach, another much simpler discrimination task, we came up with a number of 0.2 cycles per degree, which is still at I think that is a factor of 2 worse than normal mouse vision. So normal mouse vision, I would say, is kind of 0.4. And this is based on the following prediction: when we take all the cells that we record and we calculate different indices based on firing rate or based on reliability. I explained reliability before. So the blue dots are from the CMOS recording. So the reliability in this case only increases significantly with a grating width of something between 150 micrometer. So here the prediction is worse. So this is, I would say, an open question at the moment, how to interpret these results. I would like to skip this figure now and just show you maybe one last figure with a temporal resolution in activating retinal ganglion cells. There is I would say this was expected, that we transform all the on and the off ganglion cells into on-type ganglion cells. So that is actually something one would need to consider in this approach. So in summary, I've shown you that the spatial resolution up for optogenetic simulation can be very high based on a certain model. So this should always be mentioned. And of course, in laboratory conditions. So when we were able to really focus our stimulus using a microscope, using an objective, very precisely on the retina. I think that is might be different for certain in vivo applications. The inferred spatial resolution depends on the evaluation method, and the temporal filters are short and again identical for all retinal ganglion cells. In the last slide, I would like to show you some unpublished results, and maybe you don't record this slide because it is unpublished.
Günther, do you want me to pause the recording?
Yes, please.
Okay, so I'm pausing it.
Acknowledge. I would like to acknowledge my wonderful team. Today, I had the opportunity to present the work from 3 PhD students who all finished very recently and all took on postdoc positions: Andrea Corna, Monjong Li, and Miriam Rey. I have worked with very good collaborators, Roland Teves in Berlin, who is important for the CMOS project, CMOS MEA project, and 2 theoreticians, Philipp Behrens and Jakob Macke at the University of Tübingen. I would like to acknowledge funding and would thank you for your attention, and I'm happy to take questions.
Okay, thank you, Günther. So let's yes, so we have some time for question. Anyone So, okay, I have another question for you, Günther. So, thank you. This was a really exciting work and a lot of new stuff coming. Thank you very much for sharing it with us. And I just want to refer again to the to two things. One is that in some of the slides you were showing the resolution between the electrodes was you said that it will be a few tenths of micrometers the resolution could be, and then you were talking about a model predicting 10 micrometer. And what do you think are how can we have eventually in a retinal implant with an epiretinal one? What do you think? Do you think that we will be able to have a sub-receptive field stimulation, and this can translate to something? And how do you get, even if it is not a physiological relevance, what do you think is the mechanism between about this sub-receptive field stimulation? What could be the mechanism of this?
Yes, thank you. So regarding this 10 micrometer and also the spacing of the electrodes, I was not, I forgot to mention, with photostimulation, we used an additional chip. So, and we projected that chip through a microscope. So there our grating can be very, very fine. So that is not restricted by the dimensions of the recording chip. This is an important information I should have mentioned. Yeah, thank you for bringing this up. Yeah, so we had, maybe I can go back to that slide where we show them, where I show them here, this patterned light stimulus came from above using, and what is not shown here is the microscope and the objective in between the DMD and the retina. So this is, I said, it's really ideal laboratory conditions. So if this would go into an implant, it would definitely require very high density photo LEDs or so, right?
Maybe I can make my question more clear. So you have a specific number of photoreceptors in the mouse retina, and definitely it's not 1-per-1 photoreceptor per one ganglion cells. It's not a fovea.
Yeah.
And you have a very high resolution of projection, which is significantly smaller than the receptive field in the retina.
Yeah.
So if you are, let's say, stimulating this part of the receptive field and the other part of the receptive field, you don't expect the ganglion cells to behave differently unless you have some other types of cells that can make this big difference and affect other cells. I mean, I'm trying to understand what is the physiological mechanism of the ability to define other, because it's more than it's smaller than the receptive field.
Yes, I do think, sorry to interrupt you, I do think that the receptive field, that the firing rate of the ganglion cell depends where in the receptive field you stimulate it. Because the receptive field has this Gaussian profile. Even a difference of Gaussian, if one is very precise, considering the inhibitor is around. But let's talk only about the receptive field center, which has a Gaussian profile, which means if you stimulate in the center, you evoke a much higher activity as compared if you go to the 1 sigma distance or the 2 sigma distance. So I think, therefore, it is definitely possible to, sorry for the birds. It's definitely possible. It's definitely possible to even distinguish based on so at a sub-receptive field level. I would say yes, because it is not square wave shaped, the receptive field.
You think this is the mechanism?
Yeah.
I also have a question, Günther. I mean, well, it's complicated, but I'll try to simplify it. So I'm not an expert in mouse retina, and to the best of my knowledge, it's more or less composed of photoreceptors that are similar to the rods in the human retina. And I'm not even sure if there is or there isn't a fovea. So I do want to ask about the possibility to generalize, or perhaps to think about the fact that these differences may transferring this information or the methods from the mouse retina to the human may be part of the fact that there are difficulties in distinguishing at let's say at the spatial level for in human vision, because I don't know if the density of the photoreceptors in the mouse retina is different between I don't know, some area in the retina versus another area in the retina. And this is the first question. And another question was related to the actual physical arrangement of the microelectrode array. Because if we think about the human retina, and again, I don't know what happens with mice retina, we know that the processing eventually is more or less centered at the fovea, and then there's like, we can think about it as ripples around the fovea. So it's not with Cartesian coordinates, but rather more like polar, log polar coding or processing. And I was thinking what would happen if these arrays, well, at least in humans, were not all centered like a grid with a grid at the fovea, but rather distributed in a non-equal manner with growing distances between the fovea and, for example, more peripheral areas in the retina, would then maybe even at the cost of having more, let's say, precise spatial resolution, people would be able to perceive and perhaps interpolate a bigger area in space despite the loss in resolution. So that's another question.
Yes, yes, thank you. So they are very, very good questions. First, indeed, the mouse retina doesn't have a fovea as a human, so it is different. I'm aware of it, and I guess most people who work in this field are aware of it. But however, we need to do we need to start our experiments. We need to do laboratory experiments. On the other hand, it's important to, at some stage, also to test the concepts on retina, which are more appropriate than to the human. In the paper, we have some results on primate retina that we obtained. So the sinusoidal stimulation works very well on primate retina as well.
Wow.
However, that was healthy primate retina. So again, one could ask how well does that translate? But at least for primate, for non-human primate retina, the sinusoidal stimulation works. We were not. We don't have much experience with primate retina, so I cannot tell you exactly where in the. We definitely didn't record it in the fovea, but where exactly in that retina we recorded, stimulated and recorded. But definitely, I would say that it's important to be repeated in non-human primate retina, and maybe the results will be different. I don't know, but at least we were able to sinusoidally stimulate. We were not. At that moment, we didn't have our setup ready to run the discrimination task. That was a bit unfortunate, but yeah, that should definitely be repeated. The physical arrangement of the chip, that is something that can in principle be modified. That's a very good suggestion. From a technological point of view, that is not too difficult, but of course it requires a new chip design, which then in the end goes with money and all the things. But in principle, I think that's a very good suggestion to consider that the retina, in the human retina, where one would like to implant it, the cells are not evenly spaced. Yeah, that's definitely a good suggestion. Yeah.
Now I remember that I've got another question.
Yes, please.
Thank you. So first of all, I forgot to say that it's amazing work, and thanks a lot for this wonderful talk. Now I have a question of the following. I mean, in the human, let's say, eye, there is some kind of, you can think about it as jitter. Even if the light source is more or less localized in the same direction, there's, you can think about the eye movements, slight eye movements, and also the fact that once a photoreceptor is activated, that photoreceptor is, let's say, inactive for a while, and so other cells, nearby cells, take over and absorb the light particles. So now the micro electrode array is kind of like fixed over the retina. There's no, even if you do these microsaccades or the drift, it's on the same location. So I know this is technically a very like science fiction, but I was thinking if there's any possibility to also apply some jitter, even if the chip in the array is in a fixed position. I don't know, I'm thinking about some, let's say, some variations in the angles of the stimulators. No, anyway, some way to apply a very subtle jitter that may overcome the issue. I mean, the sinusoidal idea is amazing, but that would even employ a spatial jitter that you currently may not have, and that might be one of the factors contributing to the decay.
Yes, that's a good point. The jitter, we did not consider here this microsaccades here, because we wanted to start with more simpler stimuli, but jitter is definitely important. With photostimulation and optogenetics, as shown here on this slide, jittering would be very simple because we have a second stimulator on top of the retina, and there we can program, just as we program on a computer monitor our stimuli, we can program the jitter. That is definitely a very good suggestion and would be worth pursuing in the future. We didn't do it, but it's definitely a very good suggestion. Yes, thank you. For electrical stimulation, we could jitter, so we because we can apply or switch on and off the electrodes sequentially. So we can jitter as well there, but yeah, I have to think a little bit. It's not as straightforward to give you a definite answer now, but it's definitely a good suggestion, and we should take it in future. Definitely, yeah. Thank you.
Thank you. No, you don't have to use any of these suggestions. Just questions, thoughts. Yeah. Thank you.
Okay. Any more questions from the audience? Maybe I have another quick question about the temporal resolution of the optogenetics. I'm not sure if you mentioned it, but what do you think is the temporal resolution of the optogenetic stimulation?
I didn't mention it. So for the retinal ganglion cells, it is about a few milliseconds. I forgot to write it down, but it's published in Miriam's paper. So she estimated the temporal filter, and she wrote down the number. It is much shorter than for visual. So maybe I have it here. When we go to the temporal, I have scale bars. So I added scale bars. You see the visual one are more like until the first peak here. That depends also a little bit on the light intensity, but in the range of 50-100 milliseconds. And for optogenetic, these are the optogenetic spike triggered averages or temporal filters. it is below 10 milliseconds. So it's very, very fast.
Because in the past it was shown that some papers show that it was less than visual. So you think that you can have a higher resolution, temporal resolution with Dr. Zeck?
Yes, yes. If it's in the retinal ganglion cells, it would be higher resolution. I'm not sure if that is beneficial. This would need to be seen. Yeah, but I would say it's higher. Yeah. Because we don't need the whole retinal processing. We just go to the retina and to the ganglion cell and activate the ganglion cell. In this case, with the channel rhodopsin in the ganglion cell.
Okay.
Yeah.
Okay, thank you very much. So let's thank again our speaker. It was wonderful talk.
Thank you.
Thank you very much, and we hope to see more results maybe in the next few years. So thank you again.
I hope to meet you and others in some of the meetings, forthcoming meetings. Yes.
Yeah.
Goodbye.
Maybe Detroit, maybe.
Thank you.
Thank you.
Bye bye. Thank you very much.